All right, so this is part 2. We've talked about one side of the card. Now we're going to hit the other side of the 3x5 card. So we're going to talk about sporulation and germination. How come that's not the same as binary fission? Well, my quick answer is because it's not. But let's go a little bit more into it. So sporulation and germination. So sporulation is to protect the bacteria and then allow the bacteria to awaken. So protect the bacteria, sporulation, germination is then it's waking up and coming out. Sporulation occurs when the bacteria got starved and it's like, oh no, I gotta sporulate now, and go into this hibernation state. Germination is when it exits that dormant stage, okay? Next, endosymbiotic. So we talked about this in the first video on the first side of the card. And so there was a endosymbiotic theory. And essentially you had bacteria and you had mitochondria and chloroplasts, and they were all living together, all hanging out. And so they say like that mitochondria is a eukaryotic ancestor, and mitochondria, chloroplast is another. But really the biggest concept had to do with that you had this bacteria and you had mitochondria and chloroplast, and they were all living together in this whole little symbiotic relationship. Symbiotic means you get along and you work together. You do things together. So eventually they grew apart. Like half of the people in the U.S., they too grew apart. And so some of the things that are similar, kind of the reason why people thought came up with this endosymbiotic theory is because there are a lot of things that are found in chloroplast and mitochondria that are also found in bacteria. And so the teacher says, hey, you need to know 3 things. So I got you. I got this for you, teach. I got you, teach. So the mitochondria and chloroplasts both live together. That's the first thing. So both have circular DNA encoding some ribosomes and proteins. I see I have a spelling error. Imagine that. And proteins. They have ribosomal RNA that encodes 70S, and we talked about that 70S earlier. They have a double membrane. So mitochondria have that double membrane, chloroplasts have that double membrane, and bacteria have that double membrane, okay? So that's it. That's all you got to know. That was easy. All right, LPS. So I put LPS as dead, but what does that mean? So essentially the teacher might give a question about, let's say you have somebody in the hospital, okay? And that person got some sort of infection. Now, the question is, like, why would this happen? How did this happen? My response was because they didn't wash their hands and they used dirty, tools to operate on, and that was per CDC. She said, great answer, but not the answer she was looking for. And what she said after this just blew my mind, right? So let me tell you. So let's say you're going to operate on somebody and you have some knives. You want those knives to be sterile. And they use what's called an autoclave. And an autoclave is where you put the tools into this machine, and it has steam, this water and steam that's at 121 degrees Celsius, okay? And that kills off a lot of stuff. So my answer was, my response is, when the autoclaving, do everything, right? 121 degrees Celsius, it's got to kill off everything. But no. So remember how we talked about that gram-negative bacteria? So gram-negative bacteria have that lipopolysaccharide that comes up that has the O antigen. That's a molecule that doesn't get killed in the autoclaving process. I know, right? Like, I couldn't believe it. I was like, what do you mean? What do you mean it's not going to kill it? But then when I thought about it, I was like, well, remember we have these extreme, what do they call them? Like thermophils and mesophils, and those can live in like the deeps of the bottom of the, you know, hot caves and tubes and volcanoes and all kinds of crazy stuff like that. So after I thought about it, I said, okay, sure, you can have stuff that can't be destroyed by heat. So I said, so Teach, if you can't destroy it by heat, well, what are you going to do? And what she said after that, again, I couldn't believe it. You know what she told me? She said, use soap and water. Straight up soap and water, and that's how you get it. Otherwise, you need to be using, if they're going to be operate on you, you know how sometimes they're in these packages, they're already in these packages? Otherwise, you need to do that. So, really, what that said to me is I'm not going to be alcohol and everything and doing all these different things, you know, using the steam thing that they have that my friend uses to clean the floor. I'm just going to use soap and water, and I'm going to get rid of everything and be fine. All right. So moving on, we talked about that NAM on the peptidoglycogen is attached to the tetrapeptide. So I'm going to skip over that. A lysozyme. So this actually got me a little hung up. Lysozyme, not lysosome. Lysozyme is one thing. Lysosome is something else. So we're going to talk about lysozyme. Lysozyme is found in tears, saliva. So lysozyme digests bacteria and target peptidoglycan. They break the bonds between NAG and NAM. NAG and NAM. They break the bonds in between NAG and NAM. So I'll pull out my little thing. So each one of these would be NAG and NAM. Remember, the NAM is connected to the long tetrapeptide. So it would break the bonds between that. That's what lysozyme would do. I hope I didn't get that backwards. And I hope I didn't get that backwards with lysosome. Lysozyme. Let's make sure about that. Okay, we're back. So yeah, so lysozyme, you can think zymes, enzyme, okay? And that enzyme digests bacteria and targets peptidoglycogen, okay? Next, tools to help identify. What does that mean? So let's say you have a bacteria. I wish I actually had a bacteria. My teacher has bacteria. This will be my bacteria. So let's say you have a bacteria. How are we going to identify that bacteria? We can use microscopic examination. We can use culture characteristics, biochemical test, and nucleic acid analysis. So what does that mean? So microscopic examination could be like you have here's a petri dish, and you put your little, you know, bacteria that grow, and they grow in these little colony cultures. So that would be, you could look at it and have a microscopic examination, okay? Microscopic examination, well, actually, better really for microscopic examination, better example would be like the gram staining and looking at it under a microscope, okay? Because culture characteristics are like when you have the petri dish and then you put all the stuff on it and it has all these different shapes and the culture grow in these different like colonies and everything. So that would be more for culture characteristics. Biochemical tests are like, so you can have these tubes and then there could be, what is it, catalase, I believe, that if it breaks down into, oh, I can't remember. It breaks down into two components, one of which is water. But the purpose is that you can have this tube, and then the bacteria goes into it, and if it bubbles, that could be an example of biochemical test. The others, like pH strips, we've used the pH strips before, so that could be biochemical test. Nucleic acid analysis is you're using like the DNA and the ribosomes in order to, you isolate a specific sequence, and you're looking at that, and so that would be the nucleic acid. All right, next, a nucleolus. I was very happy that my teacher pointed this out, because I was getting the nucleoid mixed up with some other stuff. So nucleoid, right, that's with the prokaryotes. Nucleus is with eukaryotes. And the nucleus has nucleoids inside of it. So see, again, that's why I say this. So we have the nucleoid for bacteria. We have a nucleus in eukaryotes. And that nucleus has a nucleolus. And so in a nucleus, so nucleolus is in a nucleus, and it's where ribosomes are made. And we love those ribosomes. We love that 16S in prokaryotes, and the 18S in eukaryotes, and they also have 70S in eukaryotes, and no, they have 80S in eukaryotes and 70S in prokaryotes. It's true. So we have rRNA, stable. So why do we use rRNA? Why is it stable? It's stable because it doesn't really mutate that much, okay? That's why it's stable. But if it does mutate, something's going to get mislabeled. Okay, next we're going to have to talk about crystal violet. So this has to do with the gram-positive and gram-negative, right? We talked about this multiple times already. And there are 4 steps. So let's go over the 4 steps in detail. First step, you have crystal violet, which is a primer, okay? So that's what happens when here's our bacteria, crystal violet goes on. That's a primer, okay? So that's for, let's say here's my prokaryote and here's my eukaryote. So next you're going to have an iodine that's called a mordant. So what happens is that iodine helps bind that crystal violet to the walls of the bacteria. Particularly, we care about it binding to the gram-negative. So next we have alcohol, otherwise known as a decolorizer. So here's our gram-negative. So the alcohol can actually punch holes in the cell wall, and the crystal violet can escape. So essentially, at this step, if you were to look under it at the microscope, it would be clear. And the last step would be what's called a counterstain using something called safrarin. So it's purple pink peptidoglycogen, and then the gram-negative is positive purple peptidoglycogen. Positive purple peptidoglycogen. Positive purple peptidoglycogen. And the gram-negative is pink. Okay? Alright, so what's next? So we have acid fast. So an acid fast, you're going to use that because remember how I said there's this waxy outer substance that in some bacteria, it's called mycolic acid. So these, you have something called a mycobacteria, and that's when you're going to use this acid fast on it. And they have mycolic acid and a waxy membrane. So in these steps, you ditch the iodine. You don't need it. We're going to do a primary stain, which is called carbofusins, I believe is how you pronounce that. Your second step is you're going to do a decolorizing agent, which is an acid alcohol. And then you're going to do a counter stain that's methylene blue. So if it is acid fast, all through the steps, it's going to remain blue. If it's not acid fast, then through the steps, the first step, it's going to be red. The second step with the alcohol, it's going to be colorless. And then methylene blue, third step, it's going to be blue, okay? Next. So let's say, what are the different, the scientific, what do they call it? Scientific experiment, you know, the official way to kind of look at things. First you observe, then you question, then you have a hypothesis, an experiment, analysis, and conclusion. Observe, question, hypothesis, experiment, analysis, conclusion. So that's 6. Actually, I just remembered I have a little bit room on my index card to talk about, you know, the different orders from domain all the way to class. But I'm pretty good at remembering that anyway. Like, I think for that it's like species, genus, family. So I remember my family, ordered, class, and then it's phylum, kingdom, domain. So, but I think I'll put it on there just in case I'm really tired tomorrow, because a lot of the times by the time I get into her class, I'm very exhausted. I just have, it's just hard for me at that time. So, okay, so let's talk about this. So we observe. So let's say we observe. So we observe, right? And we go, oh my, what is that? And then we can say, oh my, what is that? Observe. Then we question, is it food? No. I think it's tiles to a game. Let's do an experiment. And you come up with an experiment, and your experiment could be, you know, well, could you play with it? You know, is there anything that you could do with this game, right? So you do an experiment. I was trying to create a word for you. See, H-E. So we observed. Wow, there's a game. We questioned. Is it food? No, it's a game. Then we create a hypothesis. So our hypothesis would be, if the tiles are a game, are you able to play the game? Maybe would be our hypothesis. Then we would create an experiment. Maybe our experiment would be having different games that we could come up with, right? And then afterwards, we would do an analysis. So that's when we would write down our data, and we would talk about it. And so we write up this whole analysis. And last is our conclusion. So our conclusion would be, yes, this is a game. Yes, we can play with it. And after a certain point of it going through rigorous research and testing and all the rigmarole, it becomes what's called a theory, a scientific theory. Not a theory as in, think you know, unicorns fly in the sky theory, but an actual concrete theory, okay? So that's what that is. So next, the teacher may have us talk about what's the difference between prokaryotes and eukaryotes. It's kind of lengthy. I didn't write down all of them because some of them are second nature. So the ones that I kind of talked about was with prokaryotes, it's binary fission, they're symmetrical, it's single-celled, actually I should see what that's about, single something, circle, circular DNA. They move by cilia or flagellum. They have a nucleoid. We talked about that one. They use active transport called translocation. Has to do with what's happening in the actual membrane. Then eukaryotes, they're bigger. They're 5 to 50 UMs. And they separate by mitosis. They have membrane-bound nucleus. They're asymmetrical, which I don't know if I really thought about that. You know, like when I thought about it, I was like, okay, well, it makes sense, but I don't know if I would have gotten that right tomorrow. They have DNA with histones, a nucleus, mitochondria, chloroplast, endoplasmic reticulum with smooth ER and rough ER, Golgi apparatus, lysosomes, ADS. They move by flagella or cilia. They have a cytoskeleton, and, you know, some things have a cytoskeleton like plants, and use facilitated diffusion. Last. So I saw that there's actually 2 things messed up on my card, so this was good to do. Last, model organism. So what is that? Oh, why? I said model organisms. Why would we want to use organisms in order to do all these experiments? So the first reason, which I thought my teacher gave, which was really funny, was she said, because nobody minds killing bacteria. And I thought, true that. But more so, and I'm not going to go into the whole complexity of it, but ultimately they do a lot of the same thing as the more complex organisms. And so because they do a lot of the same things, you know, we don't want to do experiments on humans, but we can actually do experiments on these little bacteria, and they can give us a pretty good idea of, you know, what's going to happen. So that's it. I hope you guys liked my 3x5 index card. It's been fun, and stay awesome.